Hi everyone, I am Anna Bernasconi and I'm presenting the paper Empowering Virus Sequence Research through Conceptual Modeling. This is joint work with my colleagues Arif Cianacoglu, Pietro Pinoli and Stefano Ceri at Politecnico di Milano. So the pandemic outbreak of the coronavirus disease COVID-19 caused by the virus species SARS-CoV-2 has created unprecedented attention towards the genetic mechanisms of viruses. There has been a general lack of preparation in the research community in facing pandemic crisis in a number of aspects, including well-organized databases and search systems. So we responded to such urgent need by putting our conceptual modeling research at service of new problems that regard the modeling, storage, and search over viral sequences. We have previously proposed another conceptual model focused on human genomics. You see it on the left. We next developed and implemented a pipeline for genomic data integration and built a database for genomic sequences that is searchable through the GenoSurf web interface that you see on the right. Thanks to such previous knowledge in human genomics, we have been able to rapidly design a new solution which is useful in the novel context of viral genomics. So it's corresponding to a conceptual model and its related search system. As we were very new to the wide landscape of various resources, we first understood the context in which our modeling effort takes place. So we studied the existing data management and queries resources for viruses, which include a major database institution, primary sequence deposition databases, direct retrieval tools, secondary databases with additional features and tools, and integrative systems and satellite resources. Then we also studied the metadata that are typically used in various sources and search systems, most of all in the resources that you see in line 5 and 6. Then we approached our requirement collection phase. We performed extensive interviews to groups of virologists of various specialization, of which you see a list on the left. And each researcher has provided us with a viewpoint on applications of virology that serve as requirements for progressively adding relevant features to our database, as well as relevant search services to comply with their needs. These, for example, include the area of diagnosis of vaccine development and drug-resistant associated mutations. We finally conceived the conceptual model that you see in this slide. It is centered on the viral sequence concept, which is the central information, and it is described by its biological perspective, containing metadata that regard and describe the host sample and the virus species. Then it is described by the organizational perspective, which is the project responsible for producing the sequence, by its technical perspective, which contains the sequence technology and its parameters, such as the assembly and the coverage. The most important perspective is the analytical one, which includes properties of the sequence, such as annotation, such as genes, proteins, untranslated regions, and also variants, both on the level of nucleotides and on the level of amino acids. Variants are changes with respect to a reference sequence, which is chosen by the scientific community as the first one. The schema is general and applies to any virus. But here we focus on the example case of SARS-CoV-2 as it is very current. We were able to perform complex conceptual queries upon the viral conceptual model, which replicate the search result of very recent article. Hence, we demonstrate huge potential in supporting research upon viruses. Here you see the sequence structure of SARS-CoV-2. With the rectangles, we represent proteins and genes. And you can imagine mutations as points on this axis, on these coordinates, where the nucleotide or the amino acid is changed with respect to a reference sequence of the species. This model allows us to pose simple questions such as extract SARS-CoV-2 sequences from samples of US patients that present nucleotide variants in genes that codify for open reading frames. So for example, to perform this query, we involved 5 entities: the sequence, some information from the host sample and the virus, and the information on annotation, so the name of the genes and the existence of the variant. We realized a database whose logical schema is a direct translation of the conceptual schema, and then also a search system which queries such database, which is called VirusSurf. You can appreciate here the correspondence between the entities in the conceptual model and the filters available in the search interface. And the drop-down menu allows to choose specific values for each attribute. In red, there is the analytical part, which is handled separately to take into account the different cardinality of the one-to-one relationships, which are the ones of the analytical perspective. It is important to be prepared for dealing with pandemic events most effectively. We're acting in various directions to allow, first of all, queries that are useful for identifying vaccine properties. So we're building an extension of the model and of the search system that includes epitopes. Epitopes are short amino acid sequences that are recognized by the host immune system antigens. And it is very important to understand if they contain mutations because it helps the vaccine design. Then we're also targeting queries that are useful for understanding the impact of virus mutations. We're building a small knowledge base of variants that are linked to their correlation with the clinical and epidemiological impacts. Some examples are the disease severity, virus transmissibility, antigenicity, protein stability. And we get this information from recent literature papers. We're also packaging services for confidential use by hospitals and other users because we want to allow the many users that cannot share their data to use our database and knowledge base functionalities. This is part of an ongoing project funded by the EIT Digital Innovation Program. And finally, we also have a more long-term vision. We would like to link together the viral genotype, which is modeled by the viral conceptual model, with the genomic and phenotype information of its host. We have experience in human genomics and some in the clinical aspects, but so far, in the context of COVID-19, the link between the data of the virus and the data of its human host has been missing. So this is a very big project and would require intervention of many players, mostly for the sharing of human patient data. But we're thinking about it and we're looking for partnership on this. Thank you again for the attention, and I'm available for any questions you have.